Welcome to the DNA of Things, where we explore how our potential is realized, the intersection of DNA, environment, and behavior. I'm your host, Dr. Jeremy Koenig. Together, we are unraveling the science shaping humanity's future. Now, today, I am thrilled to introduce Dr. Barbara Kuemerle, a neuroscientist and educator at Case Western Reserve University, whose work bridges cutting-edge genetics research and accessible storytelling. She's the author of CRISPR, a gripping speculative fiction novel that dives into the ethics of gene editing. Prepare to uncover how science and storytelling can illuminate the promises and challenges of human evolution. In this episode, we discuss the revolutionary potential of CRISPR-Cas9, a gene editing technology, and we delve into the ethical dilemmas posed by gene editing. And we explore how fiction can educate and inspire. The power of storytelling, go figure. Whether you're a scientist, a student, or just curious about our shared biological future, you'll leave with new insights and actionable ideas. So let's dive in. Today, I am thrilled to welcome Dr. Barbara Kuemerle, a scientist and educator who bridges the worlds of research and fiction. At Case Western Reserve University, she's made important contributions in neuroscience, studying genetic factors that influence brain development and conditions like autism. As an educator, her passion shines through in her textbook, Development and Physiology, the Biology of You, where she brings complex concepts to life for her students. But Dr. Kuemerle also writes under the pen name Jennifer Handler. Her novel, CRISPR, explores the ethical dilemmas of gene editing technology in a thrilling and accessible way. So whether through science or storytelling, Dr. Kuemerle masterfully blends education with creativity, making complex ideas engaging and relatable. Dr. Barbara Kuemerle, welcome to the DNA of Things.
Well, thank you. I'm happy to be here.
We have a lot of stuff to get through. I think I've shared a little bit about me and my own passion for molecular biology, biochemistry. Here we are at this new era of creation, if you will, where we have this molecular tool set to manipulate our biology, to direct our evolution. The technology CRISPR-Cas9 making waves in the media through all kinds of applications, ethical and unethical. And, you've gone and built on your, incredibly strong foundation as an academic to go into the science fiction realm. And so, before we dive into some of the specifics that we were going to discuss, I'd love to hear about the journey. Like, how was it that you got to the time and space of, Dr. Barbara Kuemerle, in the capacity of an educator in genetics, PhD in genetics, and then, a split into Jennifer Handler, your pen name, and the development of CRISPR. So how does something like that happen?
Well, I think it was just pure excitement. So the CRISPR sequences were found actually around 1987, and I was still in college then. And we didn't really know what they were all about, just that it was conserved sequences. And usually if something's conserved in evolution, it's important. And it wasn't until later, around 2014, that Dr. Jennifer Doudna and Dr. Emmanuel Charpentier really capitalized on this system and took it from what it's not used for in bacteria. It's more of an adaptive immune system in bacteria, but they were able to engineer it to use it for gene editing. And at the time, we were doing some gene editing with zinc fingers and talon proteins and things like that, but it wasn't specific. So when this hit the news, they published a paper, I think in 2017 in the journal Science. It was a review article. And once I think people in the scientific community read that, we realized that this was really something revolutionary. This had tremendous promise. This was earth-shattering. And I saw that right away too. And I just got excited about it. and so a class I was teaching then, I'm still teaching now, is development and physiology. And really not a molecular biology class or genetics class, but development physiology. But in development, we talk about the blastocyst, the inner cell mass, embryonic stem cells. And I taught about induced pluripotent stem cells and gene therapies. And I thought, I have got to tell these students about CRISPR because this is huge. And if you can use it in embryonic stem cells or induce pluripotent stem cells, it's got tremendous promise. So I just started teaching about it and I was excited about it. But what I realized is if you asked folks that weren't really in science, have you ever heard of CRISPR gene editing? Like your neighbor on the street, they would have no idea what it was about at all. And I just thought with something that has such tremendous promise and potential, it would be important for people to be informed about it and educated about it, for a lot of different reasons. I mean, one thing with healthcare, there may be an ailment that you or a family member or someone might have that you might want to get into a CRISPR trial to, help with a health issue that you might have. Secondly, in the States here, we are in an election year right now. And some of the new treatments, there's a great one called Casgevy for sickle cell, but the price tag is $2.2 million.
Mhm.
So there was actually an article in our local Cleveland paper this summer where a young man who had sickle cell, and he was having a terrible time with it, a lot of pain. I think he's in his late teens or early 20s. He got into a Casgevy. He got to use Casgevy in a experiment, and it worked great for him. And he and his mom then went to Washington to lobby for funding. So I think as we're picking our candidates out, we might want to ask, are these candidates supporting research and development? And then thirdly, of course, is the ethical component of all of this. I think it's important to be informed about things before they happen. So you can start to recognize what some of the downstream effects can be. I really liked your recent podcast with Dr. Daniel Stickler, where he gave the example of if you perturb something you need to be aware of the downstream effects. So he talked about how people wanted more elk and they fed them. And so then they had more elk and the elk ate the trees and then the beavers couldn't cut the trees down and that affected the salmon population. So I think that, CRISPR is out there now, the genie's out of the bottle and, there's no going back. So we should be aware of it so that we can help mitigate some of the potential problems that it could cause.
Well, you hit something. I don't know how more direct you could hit the nail on the head in terms of it's important to know before. I mean, given what we've just come through with the global pandemic and, everybody, well, let's just say the awareness or at least the ability to recognize the phrase mRNA vaccine is, I mean, it's unprecedented. I don't know that people that is non-molecular biology people fully understand what that means, how exciting that could be if, this power is wielded responsibly, if we know the full on, side effects associated with it. But certainly on the surface, the idea to be one step ahead of the virus is pretty amazing. I mean, I think that, certainly there, let's just say policy leaves something to be desired in terms of how it was put into practice. But what case in point is like, well, great. So how do we learn from that? Right. And actually, if you look to CRISPR-Cas9, I mean, this was what, a few years ago, 5 years or more, wasn't it? Where actually it was used unethically in China. I think there were twins that CRISPR-Cas9, I think it was applied to the T cell receptor to make those twins immune to HIV, if I'm not mistaken. Is that correct?
That's 100% correct. And
Yeah.
those children, I think, are 6 years old now, twin
Right.
girls.
Right.
Mhm.
And that Chinese scientist ended up going to jail, didn't he?
Yes. He's out now and he's working on muscular dystrophy, but he vows not to use it in the well, I think he's using it in embryos, but he vows not to use IVF to have those embryos then become adults. But that's the latest that I've read on that. But yeah, he did go to jail for 3 years and had to pay a big fine.
Well, yeah, I read something else recently on that also. It's kind of, I just read it in passing, but I think there was like a cryptocurrency high net worth investor that actually wrote like an open letter to him, like saying that he wants to fund him, and it's these kind of bold statements that need to be done. And it's like, okay, and then that makes me leap to like another, call it, bold set of researchers, Banton and Best, isolating insulin that like from pig to solve for type 1 diabetes, which interestingly is another molecular technology that we benefit from today, and that is molecular cloning. So cloning the human insulin coding protein region from human DNA, putting that into a bacterial vector, expressing the protein and isolating it rather than having to slaughter a pig, for example. So I guess, how do we, maybe we're jumping into the philosophy sooner, but maybe authentically, how do we balance, boldness with ethics in your opinion?
Well, I think we just have to be vigilant.
Mhm.
And we are. I mean, every year there's a conference where scientists get together, and Jennifer Doudna is one important piece of this puzzle, where they talk about the restrictions and how to try and, keep an eye on things. And
Mm.
so we just have to try and mitigate things. But, there's your sci-fi. You can get someone, a bad actor, and CRISPR's really not that hard to do.
Right.
I could set up experiments that could be used for the wrong reasons. So we just need to be, I would say, hyper vigilant of this, and make sure, like with anything, that we're aware of what's going on. And people need to know about this too. I mean, not just those in power in the government, or the officials. George F. Will just penned a commentary in the September Washington Post, where he talked about people using CRISPR bioweapons for pathological viruses. And
Sure.
it wasn't his thought. He was quoting a foreign affairs publication, where made up of scientists that, are realizing that this could be used for not just like the stuff that I wrote about in the book, like eugenics, but actually could you use it for viral pathogens?
Right.
And so I think the people in power need to make sure that we're vigilant about this, and that we have people looking into all the bad things that could go wrong with this.
Well, yeah, it's interesting. Where I did my PhD was focused on this idea of metagenomics and molecular ecology of a mobile gene pool, specifically that mobilized by the integron gene cassette system, which
Mm.
actually is similar to CRISPR-Cas9 in that it has these conserved homologous regions that flank these functional genes. Well, what do these functional genes actually do? They are a form of adaptability for bacteria, make them invasive species. In, like, for example, the coral microbiome, I did a lot of research in that area, antibiotic resistance and known pathogens, and actually even some functionality in terms of moving around uh, uh, um, environmental detox genes in contaminated sites, right? So it's this region for hypervariability, hyperevolution, accelerated evolution, in the face of some extreme set of circumstances in the environment. And, I was quantifying these things using, initially the process of, metagenomics, just basically shotgun sequencing the environment and targeting specific regions to quantify the mobile aspect of the metagenome. But then we found some cool stuff, and then we're like, oh, well, hey, why don't we clone some of these things? So actually throw these hyper adaptive regions, into, vectors that allow you to, amplify those genes and actually put them into like lab model organisms, E. coli, for example. But now all of a sudden you could be making them drug resistant or pathogenic, et cetera. And so that required us to actually then start thinking about what our lab safety standards were and how we needed to kind of upgrade, from an environmental lab to level 1, level 2, level 3, pathogenic research. So that happened like pretty quick, and like we weren't actively studying pathogens, but we were enriching them from the environment. So, I mean, this stuff that you're talking about in terms of, well, in a sense, gain of function or directed evolution, I mean, it is here and we can do it relatively quickly. And it's actually not that hard.
Right.
Like undergraduate students can do this. High school students can probably do this. So, okay. So before we step into like the storytelling and the science, maybe you can share a little bit of your kind of first book, like the physiology and the biology of you. Cause I think when I read about you, you kind of reminded me of like Carl Sagan, right? It's like he wrote the Cosmos first, and then he wrote his sci-fi books after, like Contact, for example. And I think that kind of foundation in science, allows us to leap to these very interesting, far out but believable, stories in science fiction, because it is, grounded in the art of the possible. So maybe if you could share a little bit about your first book, and then we can dive into CRISPR specifically.
Okay, so the first book is called Development and Physiology, the Biology of You. And, the creative writing and writing a book, like a textbook, are two different things, really. So when you're writing the textbook, it has to be authentic, it has to be verifiable, everything has to be cited, it has to be the truth. And the reason for writing that is I teach a large lecture course for a lot of pre-health students, pre-med students. And I teach the development physiology portion. So they start off with courses in like DNA, RNA, cell biology, biochem. And, they would have to buy this big textbook that would cost hundreds of dollars. And I didn't want them to have to buy another big textbook that cost hundreds of dollars that wasn't even necessarily totally aligned with what I was teaching. So the students often asked, can you just post your notes up in your questions? And I thought, I really can't just post my notes, because my notes are just my collective consciousness of me pulling things from different articles. I'm not necessarily citing them because they're my notes. So I'm not paying attention to doing it appropriately without, like plagiarizing. And when a publisher came to me and said, well, we can help you with this. You can write a customized textbook for your course. We'll make it paperback. It won't be that expensive. It'll be kind of like a workbook. And then we can do some online things, active learning testing that will be easy, to get graded in it. I thought this was a wonderful idea. I thought this is a great idea. I'll do it this way. This way, everybody can get into my head about what I'm teaching, and it's also done correctly. All the figures are going to be cited and be in open access so that I can use them. And then for the students, I just offered it as a tool. I say it's not required reading, it's recommended. And if you would like to use this book, go ahead and use it. You don't have to buy a big textbook. If you find that the other materials that are posted on our course website are fine, the PowerPoints, the videos, all the other resources, then use those too. Or, you don't have to buy the book. And I had one student, I got kind of a backhanded compliment once where she said, your book was surprisingly helpful. And I said, well, I'm glad. I guess that it was a surprise. It was okay too, but as long as you found it helpful. So that's where I could really just focus in on the very specifics of what I was teaching. And it had to be correct and authentic and verifiable. Then in writing the fictional book, that's just a whole different ballgame. There it was really enjoyable because you could just make things up. If you wanted your protagonist to be beautiful and brilliant, you could do that. If you wanted your antagonist to be as creepy as he could be, you could make him creepy. You could develop your characters. You could add humor to it and just make it as engaging as possible. So you try to make both books engaging. When I teach, I use a lot of case studies. I put things in a lot of context so that students realize why it's important to learn. I always say it's important to learn normal physiology so that we can understand pathophysiology. So let's take a look at some different case studies and contexts. And I'm constantly updating it. With when COVID came out, we wanted to talk about COVID because it was such in the forefront and it had such relation to physiological processes that we studied. Now COVID's kind of getting to be somewhat of a thing of the past. And we're talking, when we do some extra work, we're talking about the GLP-1 inhibitors, because that's kind of all the rave now too, with the Ozempics and things like that. So I want to keep the course interesting and engaging, because I think we know that, if you want to learn, you have to care. And if it's engaging, you're going to make more connections in the brain.
So we talked about the first book there, Development and Physiology and the Biology of You. And the way you describe that book, the reason why you wrote it, it actually reminds me of a first-year chemistry professor that I had. His name was Dr. Owie, and he was amazing. And he was actually of that philosophy also. Like, you didn't have to buy a chemistry textbook, because he sounds to me like he might have plagiarized or he didn't go through the process of, say, a formal publication, but he basically took all of his notes and put it into a bound binder. And, if you just followed along and did the practical questions, and these are, first year chemistry, it's as hard as, I suppose, your lack of foundation is.
Yeah.
So, I mean, and that's a mixed bag depending on what high schools folks are coming from. But it's university. There was all the changes, so it's hard for first year students generally. But like if you follow that book, it would be hard to get anything less than an A- in that class. Because in terms of learning, there so many aspects that the student needs to balance, right? And like just even outside the subject matter, like what's their reading and comprehension like? Like what is their ability to, be proactive and find the right resource that complement the source material. And, I find that there were, some teachers who didn't necessarily make that easy. I think some like to make it hard also
Yeah.
for students. And, I actually remember when I first started teaching as a TA and in, is it a third or no, I was in grad school at this point. I would actually give the students like variations of the exam questions
Mhm.
um, because immediately all the anxiety would go away. Is this going to be on the exam? Is like, look, here's 500 questions. From those 500 questions, 50 of them are going to be on the exam. Do all the questions, and you'll be fine. Now you can kind of
Mhm.
listen and engage and take in this material, in a way that's kind of analogous to storytelling, right? And so there is like that aspect, I think, of your book, CRISPR, that makes a very complex topic like gene editing accessible to the masses, because especially you're bringing in, as I say, the storytelling element, the emotional aspect, and so like I think it really energy and motion, it really moves people to understand. And so I wonder if you could describe the experience of writing that piece of fiction. I know you kind of touched on it a little bit there, but I mean, what was it like in terms of even the energy that moved you to write versus, a textbook? And then, what was the publication process like? What were the different people who supported you through the process? Like, what was that like?
Well, it was a learning curve because it is different from writing a scholastic work. And I didn't know much about literature. I knew almost nothing about it. So I was very much a newbie. I did a lot of just reading, and I would look at podcasts or things that were on the internet that I could find. And what I realized is I needed to really start going to some conferences and to get some training on creative writing because I didn't have that. And it's a different form of writing.
Mm,
And that's where I really kind of learned the ropes, and that there are certain things that are required in fictional writing that you don't even know or think about. So, one of the conferences I was at, I know they talked about just the length of a contemporary fiction book. Like, really, if it's not 80,000 words or above, you're dead in the water. No literary agent's even going to look at you because a book should be at least that big. Now, I suppose if you're, John Steinbeck and you want to write up Mice and Men, once you become a good author, you can do whatever you want. But for starting out, there were some real important things that I didn't know. Like it, and I started out, I wrote it was about 70,000 words, and when I was getting a lot of rejections from literary agents, I realized I need to follow the rules of the game. And I went back and wrote another 20,000 words to get it to what was an acceptable, length for the book. And just, there were just getting critique. And I guess that's where the overlap is with science, right? I mean, when you write a science paper, you send it out for peer review and you're going to be critiqued by it. And you're going to have to maybe change some things, do it differently. And it was the same with this. It was important to send it to an editor, get the editor's input on. I had a great editor. I just hired an editor myself because I self-published it, and she gave me some really great ideas about developing the characters, about things that I was just missing that I would go back and rewrite. So it's really good to, just like you do in science, when you hand it out to your peers and you send it in for peer review, you learn from the people who are the experts at it, and they give you the advice. And then my editor wouldn't tell me what to do. She would make suggestions like, I think this is an opportunity for you to capitalize on this part right here. Here's some ways you can do it. Or I don't know how you should fix this, but you need to fix this. So think about how you can fix this. And then I would go back and rewrite it. So, and it was a process of rewriting and rewriting and rewriting. Even when you write a grant proposal, I feel like you do that. I'll never forget, for my thesis defense, my PI, my mentor, said, well, you don't tell the story the way it happened necessarily, like chronologically. because I was just doing that. I'm like, okay, here's my data. First this happened, then this happened. He goes, no. You have to tell, like, you can leave things out. You have to tell it. You can tell the last part first and the first part last, but you've got to craft a topic for your seminar, and for your thesis. And it's not just telling everything the way it happened. And it's similar with creative writing too. You really have to do that. You have to craft the story, and it's not necessarily chronological the way you're thinking of it. You want to make it interesting and engaging.
And what have you found in terms of the audience? I know it's like when did you publish CRISPR?
Just August 1st, so it's only been out a couple months now.
Okay. Any feedback yet from readers?
It's getting some good reviews on Amazon,
Okay.
and and that's so interesting to me too, because one of the very first reviews, it was a good review. It was like 4 out of 5 stars, but it said something like to the effect of, "Well, even though it's kind of far fetched and it wouldn't happen, it was still enjoyable to read." And I remember thinking, "Well, it's really not that far fetched." I mean, even when I said to the editor, she said, "Well, you need to decide what genre this is so that bookstores will know where to place it."
Mhm.
And I said, "I know." I said, "What is it? Is it sci-fi? Is it contemporary fiction? Is it women's fiction? Is it, because it has all these different elements to it."
Mhm.
And she said, "Well, I'd call it, women's fiction suspense and, science fiction." I said, "To be honest with you, I think it's more speculative fiction." The
Mm.
only thing to me that's really sci fiction in it is I have them upregulate the cyclin genes to have an embryo grow to an adult quicker. I don't think you could do that. Cancer would come into play if you really started the cell cycle that quickly. You're not going to have an embryo that can become an adult in, rapid time or whatever. But some of those genes that I was picking out, I just knew. I mean, I've got like an Excel spreadsheet in one part of the book, and some of them are, for violence, like 5-HT, it's serotonin,
Mhm.
Atom 12. There's some molecules in there that we really are some of the genes that I picked. And I thought I better be a little careful with this. I don't want to give anyone the wrong idea, but I am picking some things that we know do enhance like um, muscles and protein and things that could be used for, kind of all the wrong reasons. Although things like that can be multigenic, multi traits. You can't just take one gene and all of a sudden turn someone into a violent person. But there are genes that are involved in violence, and we do know that. So there was a lot of that in there too.
And so just maybe for the benefit of the listeners, maybe you could give us just like a quick synopsis of what the book is about.
So it's about a young researcher who's in college. We pretty much, well, towards the end of high school, she starts into college, and she finds that she very much enjoys science, and she kind of stumbles upon this new technology. She's working with a PI who people kind of think he's past his prime. He's an older guy. He's just working in bacteria, and it's really not translational. So it's not the like cutting edge that people think it is. But lo and behold, just like I see CRISPR, where they found in bacteria, it was translational. You could use it with humans, and it's a very endearing story. It's heartwarming. It talks about relationships. She's close with her family, with her mother and father, and she has an older sister, so she has a nice relationship with her sister. She's got great friends. Her sidekick, her roommate, is really funny, and they're young, so they're going to school in Midtown Manhattan. So they do a lot of things that college students do. They go out clubbing, and they go to concerts, and they go out to restaurants, and she falls in love. So there's a love story component in it too. And she learns that her father is diagnosed with early onset Alzheimer's disease. And
Mhm.
being in the sciences and in neuroscience and genetics, she understands how devastating that is and how there's no cure for it. And she realizes that this new technology that she's working on could be helpful to her father. So she's really driven then to work
Mhm.
on it and get it perfected so that maybe she can use it to help him before he, gets far enough along in his disease where he's kind of past help. So she's driven to do that. And she's still kind of naive, she's thinking all scientists are doing things for the greater good. And she attends a seminar at Columbia with a world renowned scientist. And she realizes that the work that she's doing really could catapult this whole region of science into a whole new level. And so she contacts him, and she realizes that his motives aren't always for the greater good. And so now the second half of the book gets very suspenseful because basically the bad guys want to know the secret that she has to this life changing technology. And she's faced with this daunting decision. Do I destroy it so it can never get into the hands of her more powerful adversaries? Or do I keep it to try and help my dad? And then we find out that her more powerful adversaries will really stop at nothing to get this, to learn her secret. So now you see the arc of the character where she goes from being kind of a naive person to, I've got to use my ingenuity to thwart my adversaries and save my family and basically save myself and try and help all those who are involved. So the first part of the book is kind of a lot of building up and you're learning about the main character. And then the second half, as I say in the back cover of the book, everything was wonderful until it wasn't. It just takes a turn and it becomes very suspenseful. And she uses a lot of ingenuity to try and thwart her adversaries.
Yeah. But I mean, my sense of it, the book CRISPR, is that like it taps into this, timeless ethical dilemma, akin to those discussed in like, Aldous Huxley's Brave New World, or Michael Crichton's Jurassic Park. And, I wonder how much storytelling will get into, insofar as like, future authors expanding on ideas like these and others, even then embedding how like artificial intelligence could compound or is compounding the ethical concerns of biotechnology. And, even looking at, I mean, not that we need things to be even more complicated, but, you start to merge AI with genetic engineering, to address or exacerbate challenges like pandemics or aging or climate change. It really is an interesting opportunity to engage the population more broadly and make these complex topics more accessible. But at the same time, it points to, it's not just the technical aspect of the science, but it's like the, should we or shouldn't we, or what are so there's that's the other way to look at it, is there's always the benefits, but then there are the consequences. So those intended, good and bad, or unintended, right? Because I mean, you look at, again, we talk a little bit about, COVID-19 and our tendency. I'll tell it as a story. So I remember when COVID-19 really started to become something. It was before mandates were enforced, but really I think when mandates were introduced, that's when I started to get a lot of calls from, friends and family, like asking me, should I or shouldn't I be vaccinated? And I was like, well, I'm not an epidemiologist. I'm not a medical practitioner. I was like, I understand how the biochemistry works, but I can't really like answer that question. Like really, I can't give you medical advice. And then you start even thinking about the consequences. Like, well, it's my brother asking me if he should vaccinate his children, right?
Mm-hmm.
And it's like, like, I don't really know. Like, I mean, I wasn't subject to, the mandates in the way that, some say critical care response workers might be. I was
Mm-hmm.
basically working from home. In order for me to go and get vaccinated, it was really more of like an inconvenience of renewing my driver's license. Like I wasn't being forced to do it. It was just really the inconvenience of doing it. There were so many other mandates with social distancing, et cetera. And I wasn't, going and creating myself to vulnerable populations. So there isn't really the urgency, that I, might go and do it. And so, but then when people who were being, that for all intents and purposes, effectively forced to do it, and there's all kinds of social pressure associated with it, I was like, okay, well, let me start to do the research. And, then you start to look, and you're like, oh, I don't know about this indemnification clause. Like, would I buy a car that had this kind of indemnification clause associated with? So you start to do some more critical thinking. And then I called like my, professor advisors I used to work for. And, it's one person in particular. And, she said, oh yeah, well, all my family, we all got the different vaccines. My husband did Moderna, and, I did Pfizer, and we're doing like our own clinical trials. And then I was like, I remember her telling me how she at, Oxford University, they used their bare hands with ethidium bromide to like
Oh,
move around
geez.
cells. It's like that's a carcinogen, and so like I was like, oh yeah, it's like, science for the sake of science, and it's cool technically, like yeah, man, wouldn't it be amazing to split the atom just to see if we could do it, kind of thing.
Yeah.
So I started to see that, and I was like, oh wow, like okay, what are we doing here? And like, I mean, I did research on mRNA vaccines as part of my honors work. Theoretically with the HIV virus and how might that be applied. This was like 10 years ago. We're thinking about like, hey, wouldn't that be neat to always be one step ahead of this, this virus with this accelerated capability of mutating and evading, well, vaccines. So I guess where I'm getting at is you've got to make a decision, at the end of the day. And so enter storytelling again to maybe present the fundamentals, to present the heuristic of how might I make a decision. And so how do I weigh the risks? How do I weigh the pros and the cons? And, am I truly informed? Right. And so to me, it's like in the last, few years in particular, I mentioned at the top of the call, I did my kind of a combined major in philosophy and molecular biology. So, I was always fascinated by, this notion of, well, philosophies through the ages and storytelling. And I mean, even what we learn in scripture, it's like, there are elements of our humanity, like Homer's Iliad, for example, the
Hmm.
storytelling that is used to be sung through time to pass lessons on to society. And that's, how we build a culture of knowing and wisdom
Hmm.
and discernment, right? And
Yeah.
you don't have to be an expert in molecular biology and biochemistry to get there. So, I mean, that's why I was really excited to, speak to you, on this. And so I'm interested in how does your worldview change, or does your paradigm change now using storytelling as another tool, a more sophisticated tool for education? Like, where do you stand on that?
Oh, totally. So an upper level course that I teach, it's mostly seniors and it's for the neuroscience major. And it's called Human Learning in the Brain.
Uh-huh.
And some of the texts we use are by James Zoll. And one is text is called The Art of Changing the Brain, Enriching the Practice of Teaching by Exploring the Biology of Learning. And there is neuroscientific data on this that when you when you learn something, the more connections you can make, the more I mean, when you learn something, that's a change. It's
Right.
synaptic outgrowth. It's
Right.
neuroplasticity.
Right.
So if you are engaging people in maybe in active learning where you're stimulating for long-term potentiation and growing out synapses and you're making connections, you're going to learn that better. And
Yeah.
if it's important to you, there's a whole section we talk about emotion in learning. If it's
Right.
something that affects your emotional system as well as just your straight hippocampal memory regions in the brain, it is going to stick with you later. And in my studies with learning retention, what we found was it had to do with repetition. Now there's things called spaced repetition where you repeat over and over again. And again, that's how you go from early long-term potentiation. This is Eric Kandel's work. He got a Nobel Prize in 2000 on LTP and memory. So there's a real scientific basis to storytelling, and in the sense that I think about it is that what are you doing when you're storytelling? You're making more connections in the brain. You're bringing in emotional connections, and you're creating more dendritic outgrowth, right? I mean, there's a real science to all of this. So I think that the more we can do that, where we can invoke emotion and having people care about things, importance, salience. That's the term in psychology. If it's salient, you're going to repeat it. You're going to do it. If it's something that brings you joy or happiness, that's another thing Dr. Zoll talks a little bit about. If it's something that you enjoy doing, you'll do it over and over again. But if you have the mindset that you hate it or you hate going into this class, but if you walk into the class and you think, I wonder what we're going to learn today. It's just, and I think that like with CRISPR, that's what I wanted to try to do, is to bring the science in with a lot of other things, with emotion, with joy, with suspense, but to really kind to stimulate a lot of different parts of the brain, so that when they go back and think about this, the people who read it, hopefully they'll learn a little bit of science wrapped up with all of these other things. When I talk about Prometheus, I throw some Greek mythology in there. There's a lot of different aspects, and different people pick up on different things. That's the other thing I'm finding doing these book signings and I've got social media up now and I'll get comments on it. Uh, one person will like the science in it. The other person likes the relationships in it. They have so many different things. Like someone's described it as, oh, it's a story about, a young woman and the relationship. She's got a good relationship with her sister. Someone else, this was a review I got. He talked about the abuse part of it, how the villain scientist kind of like hits on the young female scientist. And I was like, wow, he took that to a whole different level than I even intended it to. Like me reading the book, that was not something forefront. That's just kind of something I slipped in there. But every person kind of makes it their own. And I just think that that's how we learn too. Ownership. Once you make it your own, how do you know if you learned something? If you can own it and you can explain it to somebody else. And that's I'm seeing more and more now as different people read it. Some friends of mine are just like, I just glossed over the science. I'm like, well, okay, if that's okay too. But I really love the book. But boy, when you started talking about AG, C's, and T's, I don't know what those are, or RNA, or tracker RNA, or Cas9 protein, or enzymes. That's just not my thing. But there's other people who just dove into that, and they thought that was incredible. They loved the science part of it.
But
So,
I bet you people who maybe didn't embrace that as much as, say, a more technical mind, I bet you now if they were to hear it on the news, they'd be like, wait a minute, like I read that in CRISPR. I know what they're talking about. And so they can remember it in real time when it actually becomes relevant, right?
yeah, and that's happening already. I'm hearing that from people where they'll say, oh, a lot of times too, it'll be like biotech companies
Right.
that, are now using CRISPR technology, and nobody knew what it was anymore, but they just knew like, oh, maybe my investment strategist is telling me I should invest in this. I don't know what CRISPR is, but now they're like, oh, I think I do know what CRISPR is.
Right.
Young people know more about it, like high school seniors. It's because it's becoming part of the curriculum. But a lot of people who didn't go through school when this came up, they've never heard of it. They have no idea what it's about.
Yeah. Yeah. It's like CRISPR is now for high school what PCR was for me.
Right. Exactly.
Yeah.
That's a really good analogy.
Yeah. Well, and then look at how important PCR was for all the COVID-19 tests, right?
Yeah.
And
now how they've expanded on it. They have reverse PCR and they, all kind real-time PCR, right? I just learned PCR is PCR. Now it's, it does so many different things.
Yeah. Amazing. So, what advice would you give? I mean, you could have taken this from both angles. What advice would you give scientific educators insofar as bringing storytelling into the classroom? And then, what advice might you offer the other direction, like people who have a captive audience, and how might you insert complex topics into a great narrative?
So I think with the first part of the question, as educators, a good way to include storytelling is to use case studies.
Sure.
We can actually, talk, and I do that a lot in my physiology class, and that's what the students remember. People say when you think back to college, how much of the content do you remember? Maybe not a whole lot, but you remember the professor, the mentor that you had, or you remember things that stood out to you. So I have students, I'm going to give this away for my 216 class now, but I do a case study. My youngest son, when he was, oh gosh, about 13 years old, he's a tall, skinny kid, usually played like first base, but the catcher broke his arm or something. So he had to play catcher that day. And he's not used to playing catcher. So it's the end of July, it's hot, and he's playing catcher, and he's about in the fifth grade, and it's fifth and sixth. And so, you can have a big change in a young boy between fifth and sixth grade. So the pitcher is this big guy. And here's my skinny little son in the 5th grader. And he throws a practice pitch really hard. And my son pulls up his mitt and the dust goes in his eyes and he doesn't get it, gets hit right in the chest. And I don't know if you're familiar with commotio cordis. Back in the day, it wasn't known all that much. Now we know a lot more about it. Well, it's when you go into VFib, and he got up, fell flat on his face, and he went into cardiac arrest. Luckily, we had a nurse practitioner who was another mom. She runs over. They're starting to do what he really needed was a defibrillator. So they're bringing that over from the pool, but she got his heart going again. And luckily, they got it going quick enough that he didn't have, like a lot of kids that happens to, you have to go on beta blockers the rest of your life because you can't play sports anymore. And now it's, they didn't even know. We took him to the Rainbow Babies, to our best hospital in Cleveland, and the attending wasn't there because this time it's like midnight by the time he went to the local hospital, and he had a right bundle Blanche Brock. And anyway, it all turned out fine. They got his heart back, and he hasn't had any complications. Well, I use that as a case study because it's really good for the cardiac part of my physiology course.
Right.
And I don't tell him that it's my son.
Right.
I show this picture of a 13-year-old blonde-haired kid, with his jersey on, and I go through the whole thing. So Tom had these symptoms. What would you do? Then they did an MRI, and then they did a chest X-ray, and then they looked at his, different levels of protein. I'm drawing a blank on what they were looking at. But anyway, we go through the whole case study. And then at the end, I say, well, this is my son, Tom. And I show a picture of him, nowadays. And I've had so many students that'll say to me, I remember that. I remember that. I remember what commotio cordis is right now. I remember the physiology of it because it was such an endearing story and a personal story that, that they heard about. And that's something that sticks with them. And so I think as educators, if you can bring in case studies that are relevant, that you can talk about with real people, that things that have happened to, that's what a lot of students are going to remember. And that's based on the neuroscience that we talked about: connections, emotion, salience. For the other part of the question, like writing fiction, now how to take something really complex and try to teach it to someone who is not familiar with it. I think one good way to use it is the use of metaphors. So if you say, well, it's kind of like this, so something that they can relate to, I think that's always helpful. The other thing is keep it short. I was in another podcast recently, and we were talking about the microwave generation. Like, people don't want to have to spend a whole lot of time trying to think about things or figure it out. So if you can use a metaphor, if you can break it into bite-sized pieces, then they're more apt to understand it. And that's one of the things that I'm doing now with my social media channels, and Instagram and TikTok. I'm making some little videos. Instagram, you can only do about a 90-second one, and talking a bit about CRISPR. So like, what is CRISPR being used for? Why is it so great? And where are we at right now? And I did a little video on Casgevy, which is the one for the FDA-approved treatment for sickle cell. I did a little one on blindness and how that's the Brilliance trials and how they're starting to do that. I'm getting ready to do one on the pig kidney transplant that was done this summer out of Harvard.
Right.
And why, what they use that for. I did one on Alzheimer's, like, can you use it for Alzheimer's? It's really not well suited for late onset because that doesn't have as big of a hereditary component. It's very multifactorial, except for ApoE4. So that's a possibility. But early onset, we have the presenilin genes and the amyloid precursor protein. So those are things that we can use it for. And I try to explain a little bit about that. Even though you can use CRISPR really well in culture and in mice, it's hard to get it. The delivery is part of our problem now. How do you get it deep into the body? And so there's the whole nanoparticles and lipid nanoparticles. Well, how do you explain that to someone who's never heard of a nanoparticle or lipid particle, and why it's hard to get into deep parts of the body? So I think metaphors and just snippets, little bit at a time, is helpful.
Well, this kind of like, so you gave those different use cases. One of the ones we started with was the HIV, right?
What?
So are we basically saying we can eradicate? It's not that we eradicate HIV, but we just basically, HIV is now benign to humans. Is that like a very real possibility in our future?
So his name was, well, I'm going to butcher it, but H.E. Giant-Ku. He did it. So how do you get it in every cell of the body, right? That's the thing.
Yeah.
Now with HIV, actually, that's kind of a better one because it does affect T cells. So you could do a blood transfusion. You could use induced pluripotent stem cells to make new T cells because that's what HIV affects. That's why it's so bad. It basically affects the cells of your immune system that are going to fight it off.
Right.
It really goes to the soldiers that are going to be fighting you. It knocks them out.
Right.
So, and that's what the biophysicist from China did. It was the receptor for HIV that he rendered non-functional so that HIV couldn't bind to it.
Right.
And so far we think it worked, right? But the true test would be to give these little girls HIV and see if they could fight it off, which is totally unethical. But I think in cell culture, it works fine. And so the question is, how do you get it in? And I guess if you could recapitulate your T cells, you probably could kind of eradicate HIV with that. But, no one's done it really in a human yet. So I guess we would have to see, instead of doing what he did, where he actually used it in embryos, and then those embryos were, put in surrogate mother, are in their mother, and then
Right.
were born. Would you do a study trial with people? And I guess you'd have to infuse hematopoietic stem cells to
Right.
have the proper T cell. But that's why it works so well with the blindness one, because they can inject right into the retina. They don't have
Right.
to worry about a systemic thing. They can do it in the retina.
Right.
And there's a study, actually, I have some colleagues, Mitch Drum at Case, that's working on it for cystic fibrosis. And they're trying to hit the lung stem cells. They're actually using lipoproteins to do that. But you just got to get it into the body.
Mhm.
So you can't, I think right now what we're going for is a low hanging fruit.
Sure.
Injections right into the eye, blood transfusions, stem cell transplants that we're already doing. But, it's going to get tougher the more we go. Although I was just at the Society for Neuroscience conference a couple weeks ago in Chicago, and it's mind boggling what they're using it for. They're starting to use it for platforms, for libraries where they can, we got 23,000 genes, and you can make up a library and pick out the ones that are going to be affected, that are going to be corrected with CRISPR. And that gives you a whole new pathway of learning new information. So the field is just exploding right now. And with AI, like you say, it's just making it even more. I just think there's so much promise for this, for healthcare and science and technology and just learning.
Right. Well, okay. So, and then, you fast forward and you imagine all of, say, the risks of the downstream side effects are gone. Right. And then, you've got carte blanche. It's whatever you want. You remember the movie Gattaca with Ethan Hawke?
Yeah.
Yeah. So, I mean, that comes back to the whole Aldous Huxley Brave New World is you actually have classes of society that you're building, which, is not far away from, the Aristotle's dream or Plato's vision of utopia, let's say.
Mhm.
And
Or Adolf
so,
Hitler's.
well, exactly. So that's where it's
Right.
is, this notion of eugenics, this notion of good genes, right? So, I mean, I think we're, kind of, spoiler alert, we're answering the question before I even ask it, which I think is, so you've got to lay the groundwork where the future is unpredictable in terms of what environmental conditions we might encounter. And so to go ahead and perfect our biology based on all known circumstances in the past doesn't necessarily set us up for the future we think we want. So, okay, what's the balanced answer then, or the harmonized answer then to, I think the question that's at the core of your book, or say the statement that's at the core of your book, is just because we can, doesn't mean we should. So where are you now in terms of, because I think writing allows us to think through what and discover what we think. And so given that you've written, nonfiction and you've written fiction, where do you stand now on that question? Just because we can, does it mean we should?
Well, clearly, because there's so much promise for this, I think we definitely should with
Right.
CRISPR. But
Right.
we've just got to, like you were describing with your work, we have to think through it. We have to try and, I know that, I mean, that's the beauty of evolution, right? Things are changing. That's why we're surviving,
Right.
right? Because we can't always predict the future. And with AI, maybe we can kind of predict it again. But that's why one of my favorite scientists is Jane Goodall, and she wrote a book, A Reason for Hope, and she was a primatologist. And she talked about when the bombs were dropped on Hiroshima and Nagasaki and how it just completely annihilated everything. And then lo and behold, years later, plants started to grow up again. So that's your evolution. Like, that nature is evolutionary, going to make the changes to adapt to things. But that doesn't mean that I totally go out and pollute my river right now and say, oh, well, nature will take care of it. Right. We need to think through things as much as possible so that we can mitigate the negative things while we're working on the positive things. And this is something that Jennifer Doudna realized right away. I mean, she published a book called A Crack in Creation, Gene Editing and the Unthinkable Power to Control Evolution. I think in 2017, she was realizing right now that what she and Dr. Charpentier had discovered did have these possibilities for some really negative consequences. And I don't think that she regrets her discovery. I think that she just realizes that we need to just be careful and contain it as much as possible.
Right. Right. Okay. There were 3 questions that I mentioned at the top of our conversation that I was going to circle back on. So question number 1 is, if this was your last podcast, what message would you want to leave? So, we've kind of been saying it without saying it. So let's see if we can tighten up the thesis. What is it that we want to communicate to individuals?
So I think with the book CRISPR in particular, and with learning and education, I just go back to another one of my favorite authors, Dr. Seuss. A quote that I like is, he says, "The more that you read, the more things you'll know. The more things that you learn, the more places you'll go." So education is really a foundation for progress, and I think it's important for people to continue to learn, and whether that means reading fictional books where they can learn, going and having a formal education, or just staying an educated person. I think back to my dad. He never went to college. Learned to be a carpenter. So part of the book is kind of my collective consciousness of him. But growing up, he read everything he could. He would read the Reader's Digest. We would watch 60 Minutes every Sunday. He would read magazines. He would talk to people. He didn't have a PhD, but he always kept learning. And I think that for all people, that's what I would recommend. I think that would be the message of the book. Just keep reading or learning or attending talks or looking at interesting information that's in the media. I think that's I guess one of the messages that I would want to get out. Find a way to help you learn.
Yeah. And I guess who needs to hear this above all? Like what person or what group of people needs to hear this above all others?
If you're just going to say what one person would need it, I would say, in the States, we're getting ready to elect our next president. I think it's someone in power that has to realize that this is an important technology. And I need to, maybe the president themselves doesn't need to be overseeing things, but they need to facilitate, a group of educated people that can oversee this and make sure that there are regulations in place and it's not misused. I think that's a very important group that needs to know about this.
And last question, who would you nominate to be on a future episode of DNA of Things? Who needs to be on the DNA of Things?
My students. I think a young person. I think you if you had 22-year-old, that's getting ready to graduate. I was thinking of one of my students that was just in my office yesterday. She's a dual major, a BS in neuroscience, a BA in psychology, and she's getting her master's in bioethics.
Ooh.
So, and she's going to take a gap year, and then she wants to go get an MD PhD, and she's doing great research out of one of our top labs. I think you should tap into the young minds. I think, us older people can pontificate all we want, but talk to a 22-year-old who is immersed in this right now, who's been brought up with, AI isn't new to them. CRISPR is, it's just all part of it. And I think that would be a really interesting person to talk to, is to get a perspective of someone like this, who's very knowledgeable and well-rounded, but is young, and it's their future that is, that we're making right now.
That is a very unique nomination. Very interested to in any and all folks that you would like to bring forwards. Let's connect on who that might be.
Okay.
Okay. Well, Barbara, I can't thank you enough. I'm obviously, blessed to receive my own copy. So, highly recommend it to listeners, whether you're high tech or high touch. There's, lessons in there that, well, I think we're going to be seeing decades and potentially centuries into the future affecting let's just say the next stage of human evolution. So thank you so much, Barbara, for your time and all your hard work and contribution to the field.
Well, thank you. And I feel the same way about you. I think what you're doing is really important to get these messages out to people about how to stay informed and educated, because it's important that we do that.
Wonderful. Thank you so
Yeah.
much.
Yes, thank you.
Thank you for joining us on this enlightening episode of the DNA of Things. Dr. Barbara Kuemerle reminded us that learning, whether through science or storytelling, is the foundation of progress. From the promises of CRISPR-Cas9 to the critical ethical questions it raises, today's conversation was a step forward toward understanding how we can shape a better future. In this episode, or if this episode sparked curiosity or inspired you, please share it. Share it with friends, share it with family. We are building this conscious community, Creative Commons, where knowledge is power, and together we can expand its reach. Until next time, stay curious and informed and keep exploring the DNA of things that make us who we are.